Dear students, a very warm welcome to all of you. In the previous lecture, we discussed about the role of enzymes in plant disease development. In today's lecture, we will be discussing about the role of toxins in the disease development of plants. We all of know that living plant cells are complex systems. Those complex systems where many interdependent biochemical reactions are taking place every time, and that too in a well-defined succession. These biochemical reactions, they result in intricate and well-organized processes that are essential for the life. If there is any disturbance in any of these metabolic reactions, it causes disruption of the physiological process and ultimately leads to the development of the disease in the plants. There are various factors which induce such disturbance. And among these factors are the substances that are produced by plant pathogenic organisms and are called as toxins. In today's lecture, we will be discussing the role of the toxins. First of all, let's have a look on the definition of the toxins. The toxins are defined as any substance which is produced by a pathogen and is injurious to the host, and it directly or indirectly plays a crucial role in the pathogenesis. These toxins, they directly act on living host protoplasts, either seriously damaging or killing the cells of the plant. The toxins are extremely poisonous substances and are effective at very low concentrations. So the question arises how the toxins influence the host cells. The toxins, they injure the host cells either by affecting the permeability of the cell membrane or by inactivating or inhibiting enzymes. In addition to this, there are some toxins that act as antimetabolites and induce a deficiency for an essential growth factor. The toxins are low molecular weight, non-enzymatic pathogenic factors. The toxins may be produced by any plant pathogen, including fungi, bacteria, nematodes, parasitic angiosperms, etc. The toxins are small sized, they are highly mobile, and may reach to subcellular levels of the host. As discussed earlier, the toxins, they disturb the metabolic reactions that ultimately lead to the development of a disease. These are very poisonous and are effective at even low physiological concentrations. So what are the criteria based on which we can call a substance as toxins. These criteria were formulated by Koch and are as number first is the compound. It must be isolated from the diseased plant. This compound, it should not be present in the healthy plant. The compound must be chemically characterized. And if this isolated compound is reintroduced to a healthy host, it must produce the original symptom. If any compound satisfies all these criteria, then only it can be considered as a toxin. There are various classifications based on different parameters. Number first classification of toxin is based on the source of origin. This classification was proposed by Wheeler and Luke in 1963. According to them, based on the source of origin, toxins are divided into three broad classes, including pathotoxins, phytotoxins, and Vivo-toxins. Now coming to first, that is pathotoxins. Those toxins which play a major role in the production of a disease and are responsible for the production of all or most of the symptoms which are characteristic of a disease in susceptible plants are called as pathotoxins. These pathotoxins are produced by pathogen during the pathogenesis process. The examples of the pathotoxins include Victorian, which is the causal agent of a common disease, what we call as Victoria blight of oats. Number second class of the toxins is phytotoxins. Those substances which are produced in the host plant due to host to pathogen interaction, but for which a causal role in disease is merely suspected rather than established. That is, we suspect that this substance, it may cause the disease, but it is not yet established. Those toxins are called as phytotoxins. These phytotoxins are usually the products of parasites which induce few or none of the symptoms caused by the living pathogen. You have to take care of this point, that whether all the symptoms or most of the symptoms are caused by that toxin or not. If the few or none of the symptoms are caused by the living pathogen, by its products, or its role in disease is merely suspected, then we call those substances as phytotoxins. The phytotoxins are non-specific in nature, and there is no relationship between toxic production and pathogenicity of disease-causing agents. The examples of phytotoxins include alternaric acid produced by Alternaria solanae and pyrrolurium, which is produced by Pyrrolaria oryzae. The third important class of toxins based on the origin is vivotoxins. Vivotoxins are defined as the substances that are produced in the infected host by the pathogen or its host, but which functions in the production of the disease and is not itself the initial inciting agent of the disease. That is, the disease is not initiated by these substances, but may later on function in the production of the disease. It may be produced either by the pathogen or its host as well. Example of Vivo toxin is fusaric acid, which is produced by wilt causing fusarium. Another classification of toxins is based on the plant parasite interactions. Whether the toxins are effective against all the plants or the specific plants. Based on that, we have got two classes of toxins. Number first is general toxin. Number second is specific toxin. The general toxins, they are also termed as non-host specific toxins or non-selective toxins because they are not restricted only to a specific host. They may attack a wide range of species of the plants. That is why they are called as non-host specific toxins or non-host selective toxins. As I said, that these general toxins, here the specific pathogen in which these toxins are present, they can produce in any host species. The examples of the general or non-host specific toxins are wildfire toxin, it's also known as trap toxin, 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 and cercosporin. Now coming to first class of general toxin, that is wildfire toxin or trap toxin. This toxin is a general toxin and is produced by bacterium called Pseudomonas syringae, the pathover tobacco. This pathogen, it causes wildfire disease in tobacco. If this toxin is isolated from the bacterium and is inoculated in other plants, it produces the similar diseases. That is why it is a general toxin. As far as the composition of this toxin is concerned, it is composed of two amino acids. As you can see on your screen, the structure of toxin. It's composed of threonine and toxin. The toxin as a whole, it is not toxic to the plant. If it reaches the cells of the host plant, it is hydrolyzed. Upon hydrolyzation, it releases toxin. It is basically this toxin which negatively influences the host plant. Basically, the toxin inactivates the enzyme glutamine synthase. We know in the nitrogen metabolism, there are different enzymes. We have got glutamine synthase, GOGATE, NR, NIR. Glutamine synthase has an important role in the formation of glutamine. Now this toxin inactivates the enzyme glutamine synthase. As a result, there is decrease in the level of glutamine level in the host plant and the accumulation of ammonia in the cells. As a result of the accumulated ammonia, uncoupling occurs between the photosynthesis and photorespiration. Besides, the ammonia, which is produced as a result of toxification by toxinine, it destroys thylakoid membrane of chloroplast, leading to the chlorosis and finally necrosis. So here on the screen, as you can see the effects of toxin. It inactivates glutamine synthesis. It results in the accumulation of toxic ammonia. It induces the uncoupling of photosynthesis and photorespiration. It destroys the thylakoid membrane chloroplast, and it ultimately results in the chlorosis and necrosis in the host plant. Now, coming to the second example of general toxin, that is phytotoxin. This toxin is produced by the bacterium Pseudomonas syringae and the pathover fasciola. This bacterium, Pseudomonas syringae, it causes halo blight in beans and other legumes. Like the previous toxin, if this toxin is introduced on non-host plant, similar symptoms are observed. The main effects that are caused by fasciolotoxin are the reduced growth, disruption of apical dominance, and the accumulation of amino acid ornithine. As you can see, the structure of the fasciolotoxin on your screen, it is a tripeptide toxin and is composed of three amino acids, namely ornithine, alanine, and arginine. This tripeptide, it carries a phosphosulfamyl group. When this toxin is introduced in the host by the host enzymes, it's cleaved, resulting in the release of alanine, arginine, and phosphosulfamyl ornithine, what is called as PSMO. The functional moiety of the toxin is this phosphosulfamyl ornithine. This PSMO, it inactivates the enzyme ornithine carbamyl transferase. The ornithine carbamyl transferase, it normally helps in the conversion of ornithine to citrulline, which in turn acts as a precursor of arginine. So when the PMSO inactivates the enzyme ornithine carbamyl transferase, there is accumulation of ornithine with a depleted level or a decreased level of arginine in the cells. Besides the accumulation of ornithine and decreased level of arginine in the cell, the fasciolotoxin, it also inhibits pyrimidine nucleotide biosynthesis. That is cytosine, uracil, and thymine. It also reduces the activity of ribosomes, which act as a site for protein synthesis. Besides, the fasciolotoxin also interferes with the lipid biosynthesis and also changes the permeability of the membranes. Now coming to third class of general toxins, that is toxin. The toxin, it is also a general toxin and is produced by a fungi called Alternaria alternata. This fungi, it causes chlorosis in the seedlings of many plants. Unlike previous two discussed toxins, toxin is a cyclic tetrapeptide. As you can see on your screen, the structure of toxin. This toxin, it binds and inactivates proteins involved in energy transfer in chloroplast. We know there are uh, some processes occurring in the chloroplast, that is photosynthesis, where there is energy transfer between different molecules. This 10 toxin, it inhibits the photophosphorylation process in the leaves and reduces the formation of ATP. Besides inhibiting the formation of ATP, 10-toxin also interferes with the normal chloroplast development and chlorophyll synthesis, which ultimately results in the chlorosis in leaf. The exact mechanism of the action of 10-toxin is still not clearly understood. Now coming to the second class of toxins, that is specific toxins. These toxins are also called as host-specific toxins or host-selective toxins because these toxins produce the disease syndrome only on a specific host and parasite combination. For example, if a pathogen A, it infects its specific host B, then only it results in the disease development. If it infects any other plant, then there is no disease development. The examples of these host-specific toxins are Victorian or HV toxin, T toxin, HC toxin, AM toxin. Now coming to the first example of these specific toxins, that is Victorian or HV toxin. It is called as HV toxin because it is produced by Helminthosporium victoria. This pathogen, Helminthosporium victoria, it is very selective in nature and specifically infects only the Victoria variety of oats. The pathogen infects the basal portion of the host and then produces toxin. Although the pathogen infects the basal part of the host and produces toxin, this toxin is then translocated to the leaves of the plant where it causes the blight disease. If the infection is severe, it can even result in the complete destruction of the host plant. As I said earlier, the Victorian is a specific toxin. It means that if this Helminthosporium victoria, it infects any other species or even other varieties of oats, then there is production of no disease. As far as HV toxin is concerned, this toxin production in fungi is controlled by a single gene. And the expression of the gene, that is the formation of the toxin, it's controlled by the factors of the host origin. The Victoria toxin or HV toxin, it affects cell wall, increase respiration, and decreases protein synthesis in host cell. As far as the structure of Victorian is concerned, it is a peptide toxin and has complex pentapeptide cyclic structure, as you can see on your screen. The primary target of this toxin is the cell membrane of the host cells. Besides, it also affects the metabolism in the susceptible plants. Now coming to the second example of specific toxins called as T-toxins. This T-toxin, it is produced by a specific race, what we call as T race of Cochleobolus heterotropus. The pathogen, it causes leaf blight in corns. Although there are different races of Cochleobolus heterotropus, but this T-toxin, it is produced only by the T race. And other races, they differ from the T race in the ability to produce this toxin. As far as the specificity of T-toxin is concerned, it attacks only the Texas male sterile cultivars of corn. Other cultivars of this corn, they are resistant to both the pathogen and the toxin. T-toxin is very essential for the virulence. As far as the structure of T-toxin is concerned, it is a mixture of long linear polyketol derivative, as you can see on your screen. The T-toxins are linear polyketols. Their length may vary from 35 carbons to 45 carbons. The toxin, it affects the selective permeability of mitochondrial membrane. As we know, the mitochondrial membrane is selectively permeable and is essential for the synthesis of ATP. The T-toxin, it affects or damages the selective permeability of mitochondrial membrane. As a result, the ATP synthesis by the mitochondria is inhibited. Now coming to the third example of specific toxins called as HC toxins. The HC toxins, they are produced by Cochleobolus carbonum. This Cochleobolus carbonum, it causes leaf spot disease in maize. As HC toxin is a specific toxin, it affects only the specific cultivars of maize. The detailed mechanism, how the HC toxin affects or causes the disease, is not yet clearly understood. Now coming to the fourth example of specific toxins called as AM toxins. The AM toxin is produced by Alternaria alternati, which causes alternate leaf blotch of apple. As far as the structure of AM toxin is concerned, it is a cyclic peptide and exists as a mixture of different forms. Again, AM toxin is a specific toxin. It selectively affects only some of the susceptible varieties of apple. While the resistant varieties, they can tolerate AM toxin easily, even at its higher concentrations. As far as the effects of AM toxin is concerned, it causes invagination of plasma membrane, besides causing the rapid loss of chlorophyll. Besides these specific toxins, there are some other toxins which are produced by some plant pathogens and play an important role in the disease development of the plants. Number first is fumaric acid. The fumaric acid, it is produced by Fusarium oxysporium, Fusarium heterosporum, Fusarium cubensi, Fusarium moniliforme. As far as this toxin is concerned, structurally it is a 5N butyl pyridine carboxylic acid. It is a non-specific Vivo toxin. The fumaric acid, it affects the permeability of plant membranes, water intake, it depresses respiration, it can also disturb oxidative phosphorylation. Besides, it can disturb the balance of inorganic ions in diseased plants. The fusaric acid, it causes almond hull root disease. Number second is alternaric acid. This toxin, it is produced by Alternaria solani. The hosts of this Alternaria solani include potato and tomato. As far as the constituents of this alternaric acid is concerned, it is composed of a dibasic acid. This alternaric acid, it is a non-specific phytotoxin and plays an important role in wilting, necrosis, and chlorosis. Now coming to the third toxin, that is fusicoxin. It is produced by Fusicoccum amygdalae, whose primary hosts include almond and peach. Fusicoxin is a terpenoid, a glucoside of carbotricyclic terpene. It is a phytotoxin and possesses growth regulatory properties and causes stomata to open. Now coming to the other toxin, what we call as pyrrhicularin. It is produced by Pyrrhicularia grisi. It has non-specific activity. It increases respiration and growth at low concentrations and inhibits them at high doses. It may inhibit the enzyme peroxidase catalase, cytochrome oxidase, and ascorbic acid oxidase. Now coming to one more example of the toxins, that is, actually these are enough or I should go in details. There is one more toxin like lycoyacirotolmin. It is produced by Opistoma almi and causes Dutch elm disease. The other toxins include lycomarasmin produced by Fusarium oxysporium, syringomycin produced by Pseudomonas syringae, coronatine produced by Pseudomonas syringae, Pathover atropurpurea, digitotoxin produced by Pseudomonas syringae, Pathover digitis, and Thaxomyces streptomyces. So this was all about the role of toxins in the disease development. If you have got any question, you may ask me.